Hello everyone, welcome to our own YouTube channel Face the Test LifeScience. So in this particular lecture, we are going to discuss how to make notes effectively and how to make them as short as possible, okay, along with some tips and tricks, okay. The first point is that you should select maximum of 2 books for each topic or subject, okay. For example, if you are making notes for cell biology subject, particularly for cell communication topic, you can select 1 book maximum. For example, Lordish book is sufficient to make cell biology subject notes. But all the topics are not explained in very easy way in Lordish book. For example, cell signaling pathways is not very much is not explained in very much easy manner. So for that cell signaling part, you can refer another book, for example, Bruce Albert. In Bruce Albert, that cell signaling part is explained in very great manner. So maximum 2 books you can refer to study or to make notes for one subject or that topic. In some cases, you can refer maximum of 2 books and not more than that. Okay. Then before making your notes, you should know what the chapter is about and you should know the concepts behind it, and then only you will be able to identify what all things to include in your notes, okay, which are the most important points that can be asked in the exam and what points you should not include in your notes, because they will not be asked in your exams if they are in much depth, okay. So in order to identify which all things are important from reference book for that topic, you have to analyze previous year question papers. For example, you will analyze cell biology previous year question papers, then you will be able to understand which topics are or which points are frequently asked, which topic or which points can be asked in the upcoming exam, and which points cannot be asked from the reference book at all, okay, because some points are in very much depth in almost all reference books. So you will get an idea after you analyze previous year question papers before making notes. Otherwise you will get confused and you will note down almost all the things which are there in the reference books, and that is totally the waste of time, and later onwards you will get confused when you are revising things, okay. The most important thing to remember is that the common information which is present in almost all reference books will be asked in your exam. It will not happen like there is one point which is given in Lordish book and that point is not given in any other book. Then that point will not be asked in the exam. Only the common information which are present in majority of the reference books will be asked in your exam. Okay, so keep this tip in your mind, okay, and using this tip your 60 to 70% task has got easier, okay. Then you have to note down important points after you have analyzed previous year question papers, then you have written them down, then you again have to smartly identify which are the most important points from this second point, okay. Then which points can be asked in the upcoming exam, this you have to identify, okay. Which points cannot be asked in the exam because they are in much depth. Once you are able to identify these things, then you will be able to make notes effectively, okay. So it depends on your skill and how you analyze previous year question papers. Then you will get an idea about in how much depth you should read reference books and what particular part you should study and what particular part you should skip, okay. So you have to analyze the previous year question papers very much carefully and smartly, okay. So then you have filtered your points and you have skipped all those points which you think will not be asked in the questions or will not be asked in the exams, or you will skip those points in your notes which will or which are in very much depth. Okay, so these points you have filtered, and then you will try to note down those points in very short way, okay, in the form of flowchart manner, or you can make your own diagrams from those short points. Okay, and you always remember that you have to use different color pens, because most of the time when you are writing exam, you will be able to recall that this particular point was written by me with green pen or blue pen or purple pen. Okay, so this thing do happen. Okay, for example, most of the time, if for example, if you get a question in your exam that dendritic cells will express what, and if you are given options like MHC1, MHC2, both MHC1 and MHC2, neither MHC1 and MHC2, and if you have written your notes using different color pens, you will be able to recall in your exam that yes, I have written that point using purple pen or green pen. So most of the time it happens. Okay, so this is very much important. Okay, and it will be also very much interesting when you will try to memorize things, or when you try to study. Okay, so when you see different colored pens in your notes, it will be interesting for you. Okay, and you can study for longer period of time. Okay, or for longer hours. The fifth point which I personally do is, after I make my notes in shortest way as possible, I will try to read them once, and then I will try to make movie of those points in my mind. You can use animated movie characters or scientific diagrams also you can use. For example, if I want to make movie about transcription initiation, I will imagine a double stranded DNA in front of me with 5 prime, 3 prime, 3 prime, 5 prime. Okay, then RNA polymerase will come and bind at a particular site, then closed complex will be formed, then that RNA polymerase will locally unwind a section of promoter DNA. Okay, and then closed complex will be converted into open complex, due to which the template strand will be available now, and the RNA polymerase can easily add the nucleotides to the 3 prime end. So the nucleotides will be added to the 3 prime end, and the RNA synthesis will be from 5 prime to 3 prime end. So this is how you can memorize the things. And when you use this fifth point like movie in mind, you will be able to retain information for longer period of time. Otherwise, you can retain the information for maximum 7 to 10 days. After that, you will forget. But if you use this fifth point to study, you will be able to retain or recall information for longer period of time. And it will be most helpful for you when you will solve CSIR NET part C questions. Okay, you can solve those questions in least time. Otherwise, it will take most of your time to solve that one question of part C. Okay, so this is the best trick to study and to retain the information. You can use these tricks, and you can always comment down under the comment section and let me know if these tricks are helpful for you or not. Okay, I am 100% sure that these tricks will help you very much. Okay, so for example, I have shown just the 2 examples of how to make notes and how to apply the concepts. Okay, so this particular topic is from post transcriptional regulation. Okay, about depending on the levels of iron in the body. This particular topic was explained in 1.5 to 2 pages in reference book, and even it can be more than that depending on different reference books. Okay, so I have made it short as much as possible. Okay, so this content is explained in 1.5 to 2 pages in the reference books. So you can imagine how much it is compressed. It is more than 80 to 90% compressed. Okay, and from here it is all clear that the 5' UTR of ferritin mRNA has ion response element. The IRP protein will bind to IRE when the ion concentration is low. Due to this, the 5' ferritin mRNA will be protected by IRP, and due to this, the nucleases will not be able to degrade the ferritin mRNA from 5' to 3' direction, or 5' to 3' exonucleases like PNPase will not be able to work, because this IRP is giving protection to ferritin mRNA by binding to IRE. Due to this, the transferrin receptor mRNA will be protected, or transferrin receptor mRNA stability will be increased when the ion concentration is low. Due to this, there will be no ferritin synthesis. Only this part was explained in 1.5 to 2 pages in the reference books, and we have made it 80 to 90% compact. Now the questions will be asked from here only, that when the ion concentration is low, what will happen to the transferrin receptor mRNA stability? It will increase or decrease. So every information is present in this content. Obviously, when the ion concentration is low, the ferritin or transferrin receptor mRNA stability will increase. Why? Because IRP is binding to IRE, due to which the 5' to 3' exonucleases or nucleases will not be able to degrade this ferritin mRNA in 5' to 3' direction. Okay, and when ion concentration is low, then ferritin mRNA translation will be repressed, because there is no ferritin protein synthesis. So you can smartly design such content that all the questions can be covered from your shortest notes. Okay, and this is also logical, because the role of ferritin protein is to store the ion. Okay, the body will only try to store the things when the things will be in excess, but here the ion concentration is already low. Then why the body will store that low ion in our body? Okay, if the ion concentration is already low, the body will not try to store it, because it is required for the production of hemoglobin. Okay, the body will only try to store the things when the things will be in excess. Okay, so the ferritin protein synthesis will be decreased, due to which the ferritin protein will not be able to store the less ion inside it. Okay, now when the ion concentration will be higher, exactly opposite things of this will occur. Then why should we include that in our notes? It will simply make the notes huge. Okay, so we should skip that part, because we are smart enough to understand this. What will happen when the ion concentration is high? Exactly opposite things will occur. For example, when the ion concentration is high, IRP protein will not bind to IRE, due to which nucleases will degrade this ferritin mRNA in 5' to 3' direction, and there will be ferritin protein synthesis. Okay, because ion concentration is higher, now the body will try to store that excess ion. Okay, so ferritin protein will store that excess ion, and whenever the body is deficient of iron, or whenever there is iron deficiency, the body will release, or the iron will be released from that ferritin protein inside the body. Okay, so for emergency condition, the body will try to store the excess things. Okay, so there should be ferritin protein synthesis in the case of excess iron concentration. Okay, Even if you are writing your theory exams or university exam, you can simply elaborate these things. You can easily elaborate this thing and you can write down your answer for like 1 to 1.5 pages easily. Okay. And the golden tip to score more marks in theory examination is to give your own input and significance about the answer. For example, you can write in your answer that a doctor can prescribe ferritin blood test, okay, to the patient who are anemic or who are having anemia. Why? Because when the blood test will show low ferritin concentration, ferritin protein concentration, it simply means that the iron concentration in the body is low, which means the patient is suffering from anemia. So like this, you can give your own input in your answer, and in this way, none of the examiner will be able to deduct your marks in your theory exam. Okay, so you can use these tricks in your theory exams also, and let me know if you are getting good marks or not in the by posting in the comment section. Okay, the second example is shown over here. The information part you can represent in this manner also. For example, this is from immunology. Dendritic cell express both MHC1 and MHC2. FMS like tyrosine kinase ligand signal is required for the synthesis of dendritic cells. Three types of dendritic cells are there: interstitial interdigiting dendritic cells, whose role is in antigen presentation. Plasmacytoid dendritic cells are also there, whose role is in protection against viruses by producing type 1 interferon. And follicular dendritic cells are also there, whose role is not who are not involved in antigen presentation. Now if you read different books, you will get lot of information about these three types of dendritic cells, but is it required? It is not required. Okay, so after you have analyzed previous year question papers, you know in how much depth you should study, which part to study in how much depth, you already know. Then why to study these three things in detail? Okay, we should only study that information which is sufficient or which is more than enough to solve our entrance exam questions. But if in your university exam or if in your college, in your syllabus, if it is mentioned that you should study these three types, then in that case you can study them in detail. Okay, but for entrance exam you have to analyze previous year question papers, and only after that you will be able to identify what things to include in your notes, which are the most important points and which are being asked more frequently, what points to skip because that points are not important, or those points are asked in very much, or those points are in very much depth in those reference books. Okay, so slowly you will be able to develop your skills. You can practice these skills and this is going to help you a lot. This will make your study more interesting and you will be able to recall things in very easy manner. This trick is going to work in almost all subjects, making movie in mind. Okay, it will work for you most in ecology subject. It will be very much interesting when you will try to analyze the cow, tiger, lion. Okay, food web, food chain. Okay, and the flowchart manner will be helpful for you in, like, for example, cell signaling nodes, GPCR pathway, serine threonine kinase pathway, nitric oxide pathway, JAK-STAT pathway, receptor tyrosine kinase pathway, WNT pathway. Okay, plant physiology also, phytohormone signaling like auxin, gibberellin, cytokinin, ethylene. Okay, abscisic acid signaling. In developmental biology also, the diagrammatical approach will be helpful for you. For example, chick development, sea urchin development, human development, Drosophila development. Okay, in immunology part also diagrammatically you can try to analyze the signaling, antigen presentation, antigen antibody reaction. In techniques also it will be useful. In techniques, flowchart manner will be useful. The movie in mind point will also be useful in techniques chapter. You can imagine the working of particular techniques. Once you know the working of particular techniques, you will be able to apply that knowledge while solving techniques question in your life science exams. That whether SDS page can be used in purification, protein purification or not, you will be able to imagine all of that. Okay. So I hope this lecture was very much helpful for you, and please subscribe. Only few students have subscribed to my YouTube channel. I am specifically making the English videos just for you students, those who do not understand English, Hindi. Okay. So please subscribe to my YouTube channel. It is free. Okay. And the upcoming videos will also be very much important for you. So if you don't want to miss those videos, please subscribe to my YouTube channel now. It is already free to subscribe. Okay. And you can press the bell icon below the subscribe button, subscribe label, so that you will get notified when my next videos will get uploaded. Okay. And you can always post any doubts if you have in the comment section. And we have already launched a CSIR net genetics course, or it is available for, that course is for almost all life science exams. It is beginners to advanced level course. The fee right now is only 500 rupees. So you can take benefit of that. I have explained it in very easiest way, and the teaching style is very much different. Okay, I have explained the numerical tips and tricks, conceptual questions, everything I have explained. So if your genetics is weak, you are free to join. I have also given 3 full-length demo lectures in that course. Okay, so I will be providing the links in the description of Telegram groups in which more than 3000 premium quality questions are available for your practice, and they are already been uploaded. More than 3500 students have joined the Telegram groups. It is free. Okay, so you can take the benefit of that group. I will be providing all the links in the description about my Android app, Telegram and Instagram. Okay, so you can find them and you are free to join. So okay, so I hope this lecture was very much helpful for you. So see you in the next lecture. Thanks for watching.